عليكم
عليك
السلام
السلام.
والرحمة.
خلصتوا المحاضرة اللي قبل ولا قطعت عليكم المحاضرة؟
لا خلصنا
اوكي.
جزئية بس نجدولها وقت ثاني.
شلون؟
باقي جزئية من الأسبوع اللي فات
أي.
نجدولها في وقت ثاني. لا يهم.
اوكي ممتاز. كيف حالكم شباب؟ طيبين؟
خير جاك خير.
طيب، معكم الدكتور جلوي العسيري. أنا راح أتكلم لكم في المحاضرة هذه اللي هي تمتد لساعتين عن الجلايكوليسيس ليكتشر 1 وليكتشر 2. راح نتكلم فيها عن الستبس حق الجلايكوليسيس، أهمية الجلايكوليسيس، أنواع الجلايكوليسيس، إيش الفرق بين الجلايكوليسيس في الإيروبيك والأنايروبيك. برضو راح نتكلم عن الريجوليشن حق الجلايكوليسيس. هذه النقطتين هي دي راح نركز عليها في المحاضرة هذه. الساعة الأولى راح نتكلم عن الجلايكوليسيس والستبس حق الجلايكوليسيس وأنواعها، وراح نتطرق للإنرجي yield by the bioenergetic of the glycolysis. في الساعة الثانية راح نتكلم عن الريجوليشن للجلايكوليسيس. Upon the completion of this lecture, you guys should know the definition of the glycolysis, the classification and the types of the glycolysis. You will be able to compare the differences between types of glycolysis. So what are the differences between the aerobic and anaerobic? Describe the pathway of glycolysis and its control, and explain how glycolysis can operate under anaerobic condition. Identify the enzymes of glycolysis that catalyze step in which ATP is used or formed, and in which NADH is formed. Recognize and interpret the pathways of glycolysis in red blood cells. And generally, if I ask you guys, what is glycolysis? What comes up to your mind?
The metabolism of glucose.
So what do we mean by metabolism of glucose?
The breakdown
of glycolysis. So if we divided the word glycolysis, glucose, lysis or breakdown. An overview of the glycolysis. The glycolysis is the breakdown of glucose to produce energy, ATP, via oxidation of glucose. So we know that if we look at the general equation for the glycolysis, it's glucose in presence of 6 atoms or 6 oxygen. We will get 6 carbon dioxide plus 6 water and ATP. We cannot really say how much ATP we are going to get exactly, because it depends on different pathways. We have the glycolysis pathway, we have the Krebs cycle pathway, and both of them are a continuous steps for degradation or breakdown of glucose. So when I say that, you think, okay, so what can we ask ourselves and say that, okay, how many ATP produced by glucose? Is it 10? Is it 8? Is it 34? I know that in the previous year, in the prep year, you have talked about not like in depth, but you have seen that in the mitochondria, the end result of the ATP is 34. So does that include the ATP that's produced by glycolysis, or only the mitochondria or the Krebs cycle produce the 34 ATP? So we see here there are 2 steps for glucose oxidation. One step take place in the cytoplasm, and this step we call it glycolysis. The second step, the product of the glycolysis, which is pyruvate, will travel to the mitochondria, as you've seen, and then it will go to the citric acid cycle or the Krebs cycle. There are different names for it, and then there will be more NAD and FAD produced in the Krebs cycle, and this NAD and FAD will reoxidize again, so they will lose the electron that they gain with the hydrogen. So they will lose it through a process called oxidative phosphorylation. And why? This is an important question and important thing to think about. Why they have to be reoxidized?
To give energy.
Well, one thing is to get energy, but another thing is that the level of fat and NAD in the cell generally is limited. So the NAD and fat in the cytoplasm, they are only in the cytoplasm. They cannot travel to mitochondria. And also the NAD and fat in the mitochondria, they cannot travel out of the mitochondria. So if all of these NAD and fat got reduced, so how can the glycolysis process continue? There is no fat to reduce anymore, or NAD, there is no NAD to reduce anymore. And also there is no fat to reduce anymore if it's in the mitochondria, fat or NAD. So the cell has to reoxidize again these molecules in order to keep them and in order to use them again for the other or continuing reactions. Is it clear?
Yes.
Good. So an overview of the glycolysis. So the glycolysis, that we say, is the breakdown of glucose to pyruvate and lactate. So it's interesting. What's the involvement of lactate here? Is it always pyruvate and lactate? No, the lactate only produced under a specific condition, and this condition is the anaerobic condition. So the glycolysis in the anaerobic condition will produce lactate. So however, in the aerobic condition or the normal cells, not the normal cells, the aerobic conditions, the glucose will produce pyruvate, and then this pyruvate will travel to the mitochondria to give us more energy via the Krebs cycle and then the oxidative phosphorylation process. So the glycolysis are the major pathway, is the major pathway for glucose oxidation, which occurs in cytosol of the cell. You have to really know that the glycolysis takes place in the cytoplasm, not in the mitochondria. If I ask, where does the glucose get oxidized? Well, then you will say in both in the cytoplasm and in the mitochondria. However, if I said what are the processes to oxidize the glucose in the cytoplasm, then you know it's the glycolysis. Okay. It is unique in that it can function either aerobically or anaerobically, depending on the availability of oxygen and intact of mitochondria. What do we mean by that? So now someone is doing some exercise, and then, you know, with doing the exercise, a lot of oxygen will be consumed by the mitochondria in order to produce more ATP for the muscles to work more or to do the hard work. So if we think about this, the level of oxygen will be decreasing, and the muscle itself will need more energy, isn't it? Because it needs more ATP. So then the glycolysis in this way, the environment inside the muscles will be anaerobic because there is a low level of oxygen. And in that way, we can call it anaerobic, but it is different than the anaerobic in the red blood cells. Why? Why the anaerobic situation in the muscles is different than the anaerobic situation in the red blood cells?
Because no mitochondria and blood.
Exactly. Exactly. In the red blood cells, there is no mitochondria. So the environment there is anaerobic. Okay. It allows tissue to survive in presence or absence of oxygen, for example, in the skeletal muscles. A red blood cell, which lack mitochondria, are completely reliant on glucose as their metabolic fuel and metabolized it by anaerobic glycolysis. So we know for definite, for 100%, that the glycolysis in the red blood cells are anaerobic glycolysis, and because there is no mitochondria, there is a lactate production. Okay. all of this, I'm talking like Elizabeth, sometimes I'm talking from here and there, but I'm trying to connect the lectures together. Sometimes I say more information, and then we will go to this information in the next slides of on the coming slides. So another definition for the glycolysis, as we mentioned, glyco is sweet and lysis is the breakdown. It's also known by Embden-Meyerhof pathway or Embden-Meyerhof-Barnes pathway. Okay, this is another names for glycolysis. So the glycolysis in general, if we looked in here, we see how many steps: 1, 2, 3, 4, 5, 6, 7, 8. Well, it should be 10 anyway. So there must be, yes, there is one step here to phosphorylate the glucose. Okay, so the glucose get phosphorylated. This is the first step. The second step, the glucose change from glucose to fructose. So it changed from aldose to ketose, and then the fructose, a phosphate group will be added to the fructose, so it will become 1 and 6 bisphosphatase. Then this bisphosphatase will be divided by a specific enzyme. We will mention it, and this will give us two molecules: the dihydroxyacetone and glycerylaldehyde 3-phosphate. The dihydroxyacetone again will change to glycerylaldehyde 3-phosphate, and then they will continue the steps until they go to pyruvate. And from pyruvate, if it's anaerobic, they will go to lactate. If it's aerobic, they will just travel to the mitochondria. The pyruvate will travel to the mitochondria. So what's the difference, or what's the comparison between the aerobic and anaerobic glycolysis? The anaerobic glycolysis, the pyruvate end product will go to mitochondria. In the anaerobic glycolysis, because of the lack of the mitochondria, the tissue, the ATPs, it allows to continued production of ATP in tissue that lacks the mitochondria, or in cell deprived of sufficient oxygen, like during the contraction of the muscles. The oxygen is required for the aerobic. The oxygen is required to reoxidize the NADH formed during the aerobic glycolysis through the electron transport chain. We will see that the NADH can be produced through the glycolysis. However, it still can go to the oxidative phosphorylation, the electron transport chain pathway, because the outer membrane for the mitochondria is permeable. The outer membrane, and you should know it's the outer membrane, not the inner membrane. The outer membrane is permeable for the NAD. So the NAD can bind to the surface of the mitochondria, and there are 5 complex for the electron transport chain, and from there it can produce its energy in independent process. We call it the electron transport chain. The pyruvate is reduced by, however, in the anaerobic, the pyruvate is reduced by NADH to form lactate. This will allow us to regenerate of the oxidized form of a NAD for a further reaction in the absence of the oxygen. However, the hydrogen of the NADH enters to the mitochondria by the glycerol phosphate shuttle to produce 2 ATP, or by the malate shuttle to produce 3 ATP. So do you guys, have you guys studied about the glycerol phosphate shuttle and the malate shuttle?
Yes.
Yes.
Yeah, good. So we don't need actually to go through there. So you know why the glycerol will produce 2 from the NAD and the malate will produce 3. You know that?
Yeah.
Okay. So another view, I tried to put more figures to explain more to you guys how this process is going. So in here, I want you to focus on the end product. So in the aerobic glycolysis, the end product is pyruvate. However, in the anaerobic glycolysis, this pyruvate has changed to lactate, and we look that lactate and pyruvate. The lactate as well can go back to pyruvate. There is a specific enzyme to do this process, do the regulation between lactate and pyruvate. We will mention it, but in here you have to see, you will see that the NAD has been reoxidized again to produce NAD plus instead of NAD hydrogen. Okay, so when you remember last year in the biology course, you have talked about the biological membrane, and there are several types of the proteins on the membrane. So one of these type of the membrane is the carrier protein. So this carrier protein, we can say that this carrier protein is responsible for the glucose transportation. So the carrier protein, when we look at the carrier protein usually is inside the cell in the cytoplasm. However, if there is a signal from the insulin that this cell, that there are a lot of glucose outside, so in the outsider environment or the extracellular environment, there is more glucose, this carrier-dependent transport protein will go to the membrane, will fuse with the membrane, and will allow for the glucose to go inside. So why we are thinking about the gates for the glucose? Well, we can just know the process. However, what we need to know is that there are some cells insulin dependent. In order for the glucose to go inside these cells, it's insulin dependent. The insulin should be bind to the receptor of these cells and to allow them to absorb the glucose. And for example, is the GLUT4, which is present in the muscle cells, the heart cells, and the adipocytes. However, for liver, neurons, liver, kidney, neurons, red blood cells, and brain, these cells, they have their gates, which is independent from the insulin. There shouldn't be, there is no insulin receptor. Or we can say they don't absorb the glucose based on the presence of insulin, okay? So they are independent from the insulin. All enzymes required for glycolysis are present in the extra mitochondria fraction of the cells. Why?
Because the glycolysis happened in the cytosol.
Yes,
not,
take place in the cytosol. So all the enzymes are present in the cytosol. So glycolysis, when we look at glycolysis, we will look at the, the stocks, so بالعربي الأسهم. So you invest and then you get payoff. You get the profit out of this investment. So it is two steps, or we can divide it into two categories. The energy investment phase, which is the preparatory phase, and the energy payoff phase, which is the phase where the ATP will be produced and the NAD, the reduced NAD will be produced as well. So 5 steps. During the first 5 steps, 2 ATP will be consumed. However, during the last the steps from 6 to 10, the reactions from 6 to 10, there will be a production of 4 ATP. Why it's a production of 4 ATP? We have 2 glyceraldehyde 3-phosphate. We know, I showed you the figure before, we got only 1 glyceraldehyde 3-phosphate. However, the dihydroxyacetone, we said there is an enzyme, will change the dihydroxyacetone to form glyceraldehyde 3-phosphate. So each glyceraldehyde 3-phosphate will produce 2 ATP, and in total, it will be 4 ATP. And each glyceraldehyde 3-phosphate will produce one reduced NAD. That's mean in total we will have 2 NADH and 4 ATP. In the aerobic glycolysis, so reaction 1, we say that the phosphorylation of the glucose. Why we phosphorylate the glucose? Why the glucose does not directly go inside the, like,
because we need to activate it.
well, it's not just activated. The phosphorylation of the glucose keep it inside the cell. Okay, keep it inside the cells. So the phosphorylation of the cells, the phosphorylation of glucose happen with two different enzymes. The hexokinase enzyme in most of the tissue. However, in in the liver and in the pancreas, which are hepatocytes, the glucokinase will be the responsible for the phosphorylation of glucose. And in this step, we will consume energy. One ATP will be hydrolyzed to ATP to get the phosphate. So the second step, via the enzyme phosphoglucose isomerase, we will change the glucose 6-phosphate to fructose 6-phosphate. So from the aldose structure to the ketose structure, okay? And we will know why we did this later. So the third reaction, we have the fructose now 6-phosphate. So the fructose 6-phosphate, and I want you to recognize that in here, there are two arrows, one going back from fructose 6-phosphate and one from glucose to fructose 6-phosphate. So this is a reversible step. However, this one is irreversible step, okay? And this irreversible step are very, very important in terms of regulation. So focus on that. You have to know that there are irreversible steps and their regulation is very, very specific. One example of the irreversible step is from fructose to fructose 1,6-bisphosphatase. You see these, the ATP, citrate, AMP, fructose 2,6-bisphosphate. These all are the regulator for the phosphofructokinase 1. In this step, we will need to add another phosphate. So ATP will be consumed and we will get ADP. And more importantly is the phosphofructokinase 1. This phosphofructokinase 1, there are regulators to activate the phosphofructokinase 1. These regulators are the AMP, the fructose 2,6-bisphosphate. However, the inhibitor for phosphofructokinase 1 are the ATP and citrate. We will go back again for this. So, in present, after we form the fructose 1,6-bisphosphate, now it's time to divide. And this is why we change from aldose to ketose, because we want the carbon number 1 to be free of the carbonyl group. So we can phosphorylate the carbon number 1. So, and then with the aldose, with presence of the enzyme aldose, then we will have two products. The product where it has phosphate group in carbon number 1, which is the dihydroxyacetone phosphate, and the other product, which is the glycerol, which is the glyceraldehyde 3-phosphate. Okay. As we mentioned that there should be 2 glyceraldehyde 3-phosphate. So we can produce 2 pyruvate, we can produce 4 ATP, and to produce 2 NADH. So the enzyme triose phosphate isomerase will do this job by changing the phosphate group from carbon 1 to carbon 3, and also the carbonyl group will go back to the carbon 1, and this will form the glyceraldehyde 3-phosphate. You see now we have 2 glyceraldehyde 3-phosphate, we have 2 1,3-bisphosphoglycerate, we have 2 3-phosphoglycerate, we have 2 we have 2 of the second or the 2 phosphoglycerate, because in here it's 3, but then there is the phosphoglycerate mutase will change the place of the phosphate group to the second carbon, and then we will and then that one, the phosphate group will be removed, and then we will have the pyruvate. Okay. but this will be explained more in the next slides. So we have this, the glycolysis, the structure of glucose in presence of glucokinase and hexokinase, and consuming of one ATP to produce ATP, we hydrolyzed the ATP in order to get one phosphate group on the carbon number 6. So the glucose now is glucose 6-phosphate. The second step, where glucose 6-phosphate in presence of isomerase will change its carbonyl group from carbon number 1 to carbon number 2 to produce, will change from aldose to ketose to produce fructose 6-phosphate. Step number 3, in presence of the phosphofructokinase 1 and consuming or hydrolyzing ATP to ADP, we will have another phosphate group that's added to carbon number 1. So we will have fructose 1,6-bisphosphate. Step number 4, this 1,6-bisphosphate in presence of aldose, they will give us two molecules, the dihydroxyacetone phosphate and the glyceraldehyde phosphate. Now we need to change the dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So in presence of the triose phosphate isomerase, this will change the arrangement of the phosphate and the carbonyl group to produce glyceraldehyde 3-phosphate. So we finished from the investment phase. We know that we lost 2 ATP in order to add phosphate group. Now it's the payoff phase. And the first step in the payoff phase, when the glyceraldehyde phosphate, each glyceraldehyde phosphate will go through this reaction. So in presence of the glyceraldehyde 3-phosphate dehydrogenase and in presence of the NAD and organic phosphate, we will produce reduced form of NAD, which is the NADH, and we will add another phosphate to the carbonyl group to have 1 to 3 bisphosphate glycerate. Okay, so we add more phosphate group in this step, but in the same way, we produce an energetic molecule, which is NAD hydrogen, which will be used later in order to produce energy. And then the 1,3-bisphosphate, okay, in presence of the phosphoglycerate kinase and in presence of a phosphoglycerate kinase, 1 phosphate will be hydrolyzed and will go to the ADP to produce 1 ATP. So what are the enzyme? What is the enzyme that's responsible to produce or to rephosphorylate the ADP to form ATP? It is the phosphoglycerate kinase, and this will change the 1,3-bisphosphate glycerate to only 3 phosphoglycerate. Okay, we only have now 1 group of phosphate. So this group of phosphate needs also to be needs to go to the second carbon. So we will have via the phosphoglycerol mutase. So we will change the phosphate group from the 3rd carbon to the 2nd carbon, and then in presence of enolase, this will lead to produce water and produce the phosphoenol pyruvate. Okay, a water group will be removed and the production will be phosphoenol pyruvate. So the last step, which is the second step to produce energy with the presence of the pyruvate kinase, the ADP, the phosphate group will be taken from the phosphoenol pyruvate to be added to the ADP in order to produce ATP. And then we will have the pyruvate. Okay, these are the 10 steps of glycolysis. So now we know the glycolysis in anaerobic, okay, and we will Yes, Faisal.
Doctor, I have question in the step 6. Why we
Yeah.
don't lose the ATP in the 6th step?
Step 6.
Yes, step 6.
Yeah, when we use the glyceraldehyde 3-phosphate, okay, when we use the glyceraldehyde 3-phosphate dehydrogenase. So the point in here, we need to, the cells cannot go to far more ATP consuming. Okay, so this molecules, the glyceraldehyde 3-phosphate, can be phosphorylated via other way than consuming ATP. Okay, there are enzymes can phosphorylate the glyceraldehyde 3-phosphate in another way than ATP. So the cells, in order to save the ATP, will go to this process, which is the reduction of the NAD in order to phosphorylate the glyceraldehyde 3-phosphate. Okay. في اثنين رفعوا ايديهم منهم. اي عبد الرحمن.
Doctor, the payoffs phase, I only counted 2 ATP. We said they're gonna be investment of the 4 ATP.
So you need to really think about this. So first, in the step 4, we produce dihydroxyacetone and glyceraldehyde 3-phosphate, isn't it?
Yes.
Then this dihydroxyacetone phosphate changed to glyceraldehyde 3-phosphate, isn't it?
Yes.
So now how many glyceraldehyde 3-phosphate we have?
We have 2.
2. These 2, well, yeah, I didn't multiply this by 2. We should put multiply by 2. But all steps from 6 to 10 are multiplied by 2. Okay?
Okay.
All the steps and all the product.
From step 6 to 10.
To 10.
Okay.
So we can go here. So you see this is from the glyceraldehyde 3-phosphate. So from step 6 until step 10, everything is multiplied by 2. Okay?
Okay.
So the phosphorylation of the release of the phosphate from the 1,3-bisphosphatase and the phosphoenol pyruvate, both of them are super high energy compound. So they release their phosphate under a process called substrate level of phosphorylation. Okay, so what is this substrate level of phosphorylation? Well, this in here is a general talking. So the ATP consumption. So we see that there are 2 ATP consumption in the preparatory phase. However, later we produce 2 NAD, and then we produce 2 ATP in this step and produce another 2 ATP in step number 10. Okay, so in total, we will talk about this in total, how much in total will be in here. So the NAD, what you should know about the NAD, will produce 3 ATP by electron transport chain in the mitochondria. So what is the substrate level of phosphorylation? The oxidative phosphorylation. So when the phosphorylation in general is a metabolic reaction of introducing a phosphate group into organic molecule. So what is the oxidative phosphorylation? هلا عبد الرحمن.
لا من قبل نسيت
Okay. The formation of high energy phosphate bonds by phosphorylation of ADP to ATP coupled to a transfer of electron from reduced coenzyme to a molecular oxygen via the electron transport chain, which occurs in the mitochondria. So the oxidative phosphorylation take place in the mitochondria. However, the substrate level take place in the cytoplasm when there is the formation of the high energy phosphate bound by the phosphorylation of ATP to ADP or GDP to GTP, coupled to a cleave of high energy metabolic intermediate substrate. It may occur in the cytosol or also in the mitochondria. Okay, so when we think about the donation or the removing of phosphate group in the glycolysis, so it's under the process called the substrate level of phosphorylation, not via the oxidative via the oxidative phosphorylation. So the aerobic glycolysis, we see that we consume 2 ATP, and then we have 3-glyceraldehyde phosphate. When produced in one step, we will have 2 NAD, and then 2 ATP and another 2 ATP. So the glucose in the aerobic glycolysis will give us 2 pyruvate, 4 ATP, 2 pyruvate, and 4 ATP. I think this is a mistake, and 2 NADH. So when we calculate how much energy we have from this process, so the 2 NAD will produce 6 ATP. We do have from the substrate level 4 ATP in each step. Okay, the total will be 10. We consumed 2 ATP, then the net balance will be 8. Okay, so the net balance of the aerobic glycolysis is 8. The anaerobic glycolysis, we will consume 2 ATP, but because there is no but it cannot go to the electron transported chain because there is no oxygen or there is no mitochondria. So less ATP production will be than comparing to the aerobic glycolysis. So lactate is obligatory end of the product. Why? Because if not formed, all cellular NAD plus will be converted to NADH with no means to replenish the cellular NAD. So the glycolysis will stop and the cell will go to death. Okay, so we will produce 2 NAD, 2 ATP will be produced here, and 2 ATP will be produced here. But these 2 ATP minus these 2 ATP, then we will only have an end result of 2 ATP. And we will not get any benefit from the NAD, hydrogen, because it will be reoxidized in order to form lactate. Okay. This will give, in presence, it will be reoxidized by using of the lactate dehydrogenase enzyme. So the anaerobic glycolysis, when we look at them, we will invest 2 ATP, 2 NADH will be formed, and 4 ATP will be produced. However, this NAD will be going back to this NADH will the reduced NAD will be reoxidized again to their original form, which is NAD plus, and we will only have 4 ATP. This 4 ATP, 2 of them will be balanced with the 2 ATP that has been consumed, okay, which will give us only a net of 2 ATP. Is it clear?
Yes, clear.
But what is why the lactate is obligatory? I didn't.
Why the lactate
is obligatory? I didn't understand it well.
Well, because when there is no oxygen, the cell, the pyruvate cannot go to the mitochondria because it cannot do anything, because you know that in the mitochondria, in the electron transported chain, the cells will need to consume oxygen. However, the cell is already under a low level of oxygen, so they will go to lactate, and this will be a signal. Later we will see that this lactate travel to the liver in order to tell the liver that, well, there is a less, that we need more glucose, because we need more glucose because the environment in the muscles is very anaerobic, so we need more glucose in order to get more energy. Okay.
Okay.
إياد.
دكتور، في الخطوة العاشرة الكربون نمبر 3 كيف تحولت من مثالين إلى مثال؟
وش الإنزيم اللي استخدم؟
لحظة خليني أرجع. بايروفيت كاينيز.
طيب في الخطوة نمبر رقم 3 كيف
لا،
تحول
10.
من، في الخطوة رقم 10 كيف تحول لما يروح الفوسفيت وش يصير؟
دايفوسفيت، تايفوسفيت.
اوكي، صار في ترايفوسفيت، هذا للاي تي بي، الفوسفيت هذا راح للاي تي بي، صح ولا لا؟
صحيح.
وش يبقى؟
يبقى بايروفيت، صح؟
امم
يبقى بايروفيت.
يطلع لنا البايروفيت. اوكي.
إيه بس قصدي كانت CH2 صارت CH3.
دقيقة. اوكي. اوكي، هذه هنا فيها خطأ، معلش. تمام، واضح؟
واضح.
أيوة.
إياد أنت اللي رفعت يدك، صح؟
إيه نسيتها.
أي. طيب في شيء جدا مهم يا شباب، لأن أمس كان عندنا اجتماع قسم، فكان في تشديد جدا مهم بالنسبة لمرجعكم الأساسي هو إيش؟ ليبنكوت. الأسئلة ترى بتجي من ليبنكوت. تمام؟ مرة في تشديد من العميد، في تشديد من القسم على هذا الشيء. يعني ان نحط يعني انا اوكي احاول اشرح الميكانيزم اللي موجوده في ليبنكوت. تمام. لذلك اذا عندكم اي اسئله انا بحط ايميلي وبحط رقم المكتب، تجون تسالوني. تمام، لان هذه اشياء لها علاقه بالجوده وبجوده مخرجات الكليه. في طلاب من يوم ما يدخل الكليه الين يتخرج ما يمسك الكتاب، لان الاسئله تجي من السلايدات، وهذا غير جيد ابدا. تمام. So when we talk about the red blood cell. So what's the difference between the red blood cell, the anaerobic condition in the red blood cells, and the anaerobic condition in the lactate? We say that the red blood cells, they don't have mitochondria, so this is their condition, is always anaerobic. So the red blood cell dependent on glycolysis for energy. The red blood cell deliver O2, oxygen, to the body tissue by protein called hemoglobin. Red blood cells lack intracellular organelles such as nuclei and mitochondria. The mitochondria used بالنسبة لموضوع الهيدروجين هذه الثانية، خليني بتأكد منه، لأني أشوف أنا حتى هنا في الكتاب حاطين لكم. أوكي. معليش يا شباب، معليش. أنا شكرا لك يا إياد على سؤالك، لأنه شكرا لك على سؤالك، لأنه هنا المفروض يكون فيه another إيش bond. أوكي، فبتصير ما أدري كيف صارت هي الحين عندي في السلايد، لكن هنا في دبل بوند، عشان كذا هي إيش CH2. تمام، هنا في إيش، في إيش يا شباب؟ دبل بوند. تمام،
تمام.
يعني عشان كذا أنا أقول، لأن بعض الأحيان تكون السلايدات موحدة بيننا وبين الفيميل، فيصير فيه ميكس للسلايدات. يعني لو تشوفون هنا الآن، هنا في دبل بوند. تمام، هذا هو السبب. واضحة؟
ايوه، يعني CH2 بس ينتج
اي
CH2.
CH2 بس انه في دبل بوند، عشان كذا هي CH2. تمام، لاننا شلنا 2 ووتر، لان شلنا ال OH اللي موجود فيها بالاضافه للهيدروجين، فهذا ايش اوريدي راح يخلي ايش فيه 2 بوندز. الميتوكوندريا use الاكسجين to produce energy, therefore the red blood cell do not have mitochondria, maximize the oxygen ايش transferring. In glycolysis at the red blood cell, pyruvate convert to lactate. The accumulation of lactic acid in the red blood cell will increase the acidity of the red blood cells and affect hemoglobin. Lactate accumulation in liver accumulate in liver and form glucose through gluconeogenesis. Okay, so what is the 2,3-bisphosphoglycerate rate and why it is present in the red blood cells, and what is the effect of this step on the red blood cells in terms of energy? طبعا الآن الريد بلاد سيلز فيها بروتين، شو اسمه البروتين هذا؟ هيموغلوبين، صح ولا لا؟
صحيح.
الهيموغلوبين هذا يرتبط بايش؟
أكسجين.
بالاكسجين. طيب الاكسجين هذا كيف ينفك ارتباطه بالبروتين بالهيموغلوبين؟ عن طريق وجود انزيم. الانزيم هذا يتم انتاجه خلال عمليه الجلايكوليسيس. الانزيم هذا اللي هو ايش؟ ال 2,3-bisphosphoglycerate، يتحول ال 1,3-bisphosphoglycerate via الميوتيز الى 2,3-bisphosphoglycerate. تمام، البريزنس حق ال 2,3-bisphosphoglycerate بيساعد على decrease the affinity of oxygen, which favor the unload of oxygen even at low oxygen pressure. Then via a phosphate, via phosphatase, okay, an enzyme called phosphatase, and presence of water, the phosphate group will be removed. Okay, or I mean, one phosphate group will be removed, and it will be replaced by hydroxyl group, and this will give us the 3-phosphoglycerate. وش المشكلة اللي حصلت هنا؟ كيف هذا حيأثر على الطاقة اللي حتنتج؟ احنا عارفين انه من ال 1-bisphosphoglycerate، من 1,3-bisphosphoglycerate ل 3-phosphoglycerate فيه انزيم اللي هو الفوسفوجلايسريت كاينيز. الانزيم هذا راح يساعدنا في انتاج ايش 2 ATP، صح ولا لا؟ لما يصير هذا الديتور على الستيب هذه فيه 2 ATP، ما راح تنتجها الريد بلاد سيلز. طيب احنا عارفين ان في الانايروبيك كونديشن we will only have 2 ATP، صح؟
صحيح.
طيب في الكونديشن هذه كم ATP بيكون عندنا؟
ولا شيء، صفر.
0 ATP. Okay, so it is not a favorable process, but it's a process that's needed, and this could be the reason why the glucose go to the red blood cells without need for insulin. So it can directly go to the red blood cells. So the supplement of the supplying of glucose to red blood cells is not conditioned, because some of the red blood cells, some of this glucose will produce energy and some of them will not produce energy. Some of them will have another job in order to release the oxygen. but some of them, they will have another job, which is give the energy for the red blood cells. Okay. The 2,3 phosphoglycerate is an important regulator of the binding of oxygen to hemoglobin. It is the most abundant organic phosphate in the red blood cells, where its concentration is approximately that of hemoglobin. The the BPG, the 2/3 PPG, synthesized from an intermediate of the glycolytic H pathway, and we mentioned this, that it's decrease the affinity to the oxygen and favor the unloading of O2 even at low temperature. So what is the significance? We mentioned that it's unload the oxygen. It also affect in the hypoxia. So it can help to prevent tissue hypoxia in condition where it's more likely to occur in condition of low tissue oxygen concentration, like the airway obstruction, anemia, congestive heart failure, will tend to cause the red blood cell to generate more 2,3 bis phosphoglycerate in their effort to generate energy by allowing more oxygen to be released in the tissue deprived of oxygen. There is also another benefit, which is in the fetal hemoglobin, which is the blood that goes through to the babies. So the fetal hemoglobin exhibit a low affinity for the 2/3 bisphosphoglycerate, resulting in a higher binding affinity for oxygen. This increased oxygen binding affinity relatively to that of adult hemoglobin is due to the hemoglobin fetal having 2 alpha and gamma dimers as opposite to the 2 alpha and beta dimers in the hemoglobin of the adults. The positive histidine residue of the hemoglobin adult, the beta subunit, that are essential for forming the 2,3 bisphosphoglycerate binding pocket, are replaced by a serine residue in the hemoglobin fetal gamma, which is the gamma subunit. So the difference between the beta subunit and the gamma subunit in the fetal hemoglobin and the adult hemoglobin, that this subunit, instead of histidine, it will have a serine residue, and that will change it from the beta to gamma. The 2/3 bisphosphoglycerate has the difficulties in linking to the fetal hemoglobin. So the affinity of the fetal hemoglobin for oxygen increase. That's the way the oxygen flows from the mother to the fetus. So to keep this type of blood to reach to deliver the oxygen to the fetus, in order for the baby to get this oxygen, the blood will form the fetal hemoglobin, which does not favor the 2/3 bisphosphoglycerate, because if it can be response to the 2/3 bisphosphoglycerate, it will release the oxygen in the body of the mother without delivering it to the fetus. So the anaerobic glycolysis, it will produce 4 ATP, consumed 2 ATP, and there is a net of 2 ATP. This is the general. However, if there is in the red blood cells, only when first site of ATP generation is bypassed, the net balance is 0. So in summary, the glycolysis in red blood cells, or the end product, اللي هو lactate, and there is no net production or consumption of NADH, so there is no energy can be produced from NADH, and the energy yield, if there is no process or detour to the 2,3 bisphosphoglycerate is formed, there will be 2 ATP. However, if the shuttle or if the shunt or the detour of the 2,3 bisphosphoglycerate is present, we will have only 0 ATP in the cells. So the take-home message is that the aerobic glycolysis will have 8 ATP, the anaerobic glycolysis will have 2 ATP. The net, however, in the red blood cells, without the synthesis of the 2/3 bisphosphoglycerate, we will have 2 ATP. However, with the synthesis of 2/3 bisphosphoglycerate, we will have only 0 ATP. Okay. For me, I don't like to give all the lecture like once. I like to take a break. So is it okay if we take a break for 5 minutes?
Of course.
Okay. So you guys have a break a little bit, go through the previous slides. If you have any questions, if we cannot answer this question because we only still have 1 hour, I'm not sure about the regulation process. It might take more because it needs to explain more. We need to explain more. And it's a little bit complicated, not very complicated. So if you have questions, you either can write them. Do you have a share words file of words that share between you guys? So you can write your questions about my lectures. So if I'm doing the lecture and there is no time to answer your question, you can write just your questions and then just send it to me. Yazid can send it to me if that's okay. Okay. Or you can send me via email if that's more easier for you. So now you'll have a break. So we'll go back at 7 minutes after 10. 10 و7 دقائق. السلام عليكم ورحمة الله. نرجع نستكمل محاضرتنا اليوم. So far يا شباب فاهمين؟
فاهمين بس يبيلها مراجعة.
ايه يبي المراجعه.
وش الشيء اللي تحسونه صعب so far إلى الآن؟
يمكن كميه معلومات.
أوكي.
والانزيمز برضه يا دكتور.
هين في أشياء صراحة يبغى لها حفظ. هي مشكلة الجلايكوليسيس إنها فيها إنزيمز كثيرة، نفس الكريب سايكل، نفس الأشياء الثانية هذه يبغى لها حفظ يعني. لكن إن شاء الله الأمور تتيسر يعني. أوكي. لكن كمعلومة، هل مثلا الآن رسمت عندكم فكرة عن الجلايكوليسيس؟
إيه كخطوات وكمعلومة واضحة، بس يبغى
إيه.
نحفظ الإنزيمات وترتيب المواد.
طيب أوكي. Okay, the second lecture we will be talking about the regulation of the glycolysis, and before that we will mention the state which is called lactic acidosis, and when this state happen and what's the complication of this condition. We will also describe the inhibitors of the glycolytic pathway. We will also describe the regulation of the glycolytic pathway. We will identify the regulatory enzyme. The regulatory enzyme mainly in the irreversible reaction, in the irreversible step. We know that in the glycolysis, if you go back to Lippincott, you will have a general view of the glycolysis. You will see some of the step, there is only one arrow, like from glucose 6-phosphate to fructose 6-phosphate, but there is no way to change back from fructose to glucose. However, actually there is a way for that, but it is a detour pathway. So first, before we know what will happen, because this way the detour pathways to form the glucose again, we will take it and we will talk a lot about it in the gluconeogenesis in the next week, inshallah. However, this week we will be only focusing on the glycolysis. So these three irreversible reactions are the glucokinase and hexokinase, which is the first step, the phosphofructokinase, which is the step that changed the fructose 6-phosphate to 1,6-bisphosphate, fructose bisphosphate, bis-fructose phosphate, and the pyruvate kinase, which producing the kinase. If you notice, there are two of these enzymes are involved in hydrolysis of ATP, in consuming ATP, and one of them are involved in forming the ATP. Okay. And then also we will describe the regulatory mechanism. There is a rapid and short term of mechanism. It's either allosteric or via the covalent modification, or slow and long term, which is via hormones. Either in the induction, this hormone will be induced or repressed. So the lactic acidosis, which is the elevation of the concentration of lactate in the plasma, term lactic acidosis because it's decreased the pH, so it's increased the acidity of the blood, occur when there is a collapse of the circulatory system and cause myocardial infarction, pulmonary embolism, uncontrolled hemorrhage, and shock. The failure to bring adequate amount of oxygen to the tissue results in impaired oxidative phosphorylation and decrease the ATP synthesis. To survive, the cell will rely on the anaerobic glycolysis for generating ATP and producing lactic acid as the end product. In many clinical situations, measuring the blood level of the lactic acid allows the rapid early detection of oxygen depletion in patients and the monitoring of their recovery. Okay. The lactic acid may also occur in conditions in which oxidation of pyruvate is impaired, as in thiamine deficiency. We will talk on Thursday about the pyruvate and how the pyruvate is converting from pyruvate into acetyl-CoA inside the mitochondria. And there are some cofactors and coenzymes that regulate this process. So part of that, when there is an accumulation of pyruvate, the pyruvate will change because the deficiency in one of the cofactor or the coenzyme, such as thiamine, a deficiency in the pyruvate dehydrogenase enzyme, and also a poisoning by mercury or arsenic. This, the presence of mercury or arsenic will block the SH group of the lipoic acid, which is also a regulator of the pyruvate acetyl-CoA conversion reaction. If there is any problem with this, that will lead to increase of lactic acidosis. Okay, or increase the production of lactic acid because of the accumulation of the pyruvate. So the pyruvate cannot change to acetyl-CoA, so it accumulates in the cell. So the cell changes this pyruvate into lactate. So inhibitors of glycolysis, there are 3 ways to inhibit glycolysis, either by arsenate. Okay, and this arsenate is chemically similar to the phosphate. So in the glycerol 3-phosphate, the glycerol 3-phosphate dehydrogenase is susceptible to this arsenate. So the arsenate will bind to this glycerol 3-phosphodehydrogenase, and it will block the reaction from proceeding. Okay, so the arsenate can bind to the phosphate group region and inhibit the phosphorylation of the G3 phosphate and forming 1 arsenate, 3 phosphoglycerate. This will lead to not production of ATP from 3 bisphosphoglycerate and give us 0 net of ATP. So if we block this reaction, we know that the first reaction to produce energy is changing the 1,3 bisphosphoglycerate into 3 phosphoglycerate. If we block this reaction and we block this molecule, the glycolysis will not go further. It will stop. So this is why the arsenate is very important, and it also can affect the glycolysis. Another example or another inhibitor is the iodoacetate, which is an inhibitor of the glycerol 3-phosphate dehydrogenase as well. And fluoride, which inhibit the enolase. The enolase, we know the enolase is very important in through the process. So if it's inhibited by if it's inhibited by fluoride, that will not allow the 2-phosphoglycerate to change into phosphoenol pyruvate. And then the process will stop and the glycolysis will not go on. So the regulation of the glycolysis, we say that there are 3 irreversible reactions: glucokinase, phosphofructokinase, and the pyruvate kinase. The regulatory mechanism, we mentioned that they are either rapid, and the rapid and short term include the allosteric and covalent modification, which is adding phosphorylation group or removing phosphorylation group, or adding or presence of molecules. The allosteric, we mean presence of some molecules that can bind to the active site, or their presence will activate this enzyme, such as presence of ATP, citrate, ADP, AMP. The long term, which is presence of hormone, either they are bind to the receptor by inducing or repressing, because we will see that some of the enzymes, they are originally found in the nucleus, and some of this protein can manage their production. Okay. In the induction and repression, which is the change reflects an increase or decrease in the gene transcription, resulting in an increase of the enzyme synthesis. So in the regulation of the glycolysis, we have the first step, which is the hexokinase and glucokinase. We have the phosphofructokinase and the pyruvate kinase. So in terms of enzymes, I want you to know that the glycogen and insulin only regulate the glucokinase. Why? Because the glucokinase is present in the liver. And in the liver, there are a lot of metabolic processes. The liver are responsible for synthesis of glucose, as well as there is a process as well as degradation of glucose. So it's very needed in there, and it can be regulated by insulin and glucose. It's not like the hexokinase, which is only regulated by the concentration of the glucose. Also, the glycogen and insulin are involved in the phosphofructokinase 1. Also, they are involved in regulating the pyruvate kinase. The presence of the glucose 6-phosphate will be an allosteric regulator for the hexokinase. Why? Because the presence of a lot of glucose 6-phosphate will inhibit the hexokinase. Also, the presence of a lot of ATP and citrate will inhibit the phosphofructokinase, because that will tell the cell that we don't need glucose. We have a lot of glucose and we don't need any more phosphorylation, or the cell has enough energy, so it doesn't need more energy. Another allosteric inducer. So on the right side, we will have the hormonal regulation, okay, which is the slow regulation, and the left side, which is the rapid and short regulation, which is the allosteric and covalent modification. So the allosteric, we mentioned that one of them is the glucose 6-phosphate, which regulates the hexokinase. The phosphofructokinase, there are 5 molecules can regulate it. ATP and citrate inhibit the phosphofructokinase. AMP, fructose 6-phosphate, and fructose 2,6-bisphosphate all induce the phosphofructokinase 1. Also, the phosphofructokinase 1, the presence of high level of phosphofructokinase 1 increase or induce the pyruvate kinase. The ATP will decrease or inhibit the activity, will inhibit the activity of the pyruvate kinase. So this is also another figure. It says the same, that glucokinase regulated by insulin and glucagon, phosphofructokinase regulated by insulin and glucagon, and all the irreversible steps in the liver are regulated by insulin and glucagon. So the effect of the hormones on the glycolysis, the insulin induce the synthesis of 3 irreversible enzymes, dephosphorylate the enzyme through the activation of the protein phosphatase. Glycogen repress the synthesis of the 3 irreversible enzymes. Phosphorylate the enzyme mediated by the cyclin A and B activation of protein kinase A. We will look at this. So the regulation of glucokinase and hexokinase, the hexokinase is inhibited, as we mentioned in the figure. So all the reading that I will read now, I just mentioned it in the figure. So the hexokinase is inhibited by glucose 6-phosphate when accumulated. Glucokinase is inhibited indirectly by fructose 6-phosphate and is indirectly stimulated by glucose. So the glucokinase, it's a little bit different than the hexokinase. Why? Because the glucokinase regularly is present coupled with the glucokinase regulatory protein inside the nucleus. However, when there is a high level of the glucose or a high level of fructose 6-phosphate, the glucokinase will travel from the nucleus to the cytoplasm in order to induce the glycolysis. Okay? So the inactive form of the glucokinase find present in the nucleus. However, if there is a high level of glucose or fructose 6-phosphate, it will travel to the cytoplasm. This is a comparison, and it's very important table. It can summarize what we have been saying about the hexokinase and glucokinase. The hexokinase extrahepatic, however, the glucokinase in the liver, pancreatic beta cell, the substrate, all hexose, and glucose. However, for the glucokinase, it's glucose mainly. Enzyme kinetic of the hexokinase, it has a high affinity, so and it's active at low glucose concentration. which ensure supply of glucose for tissue with a low velocity, and the low affinity. However, for the glucokinase, it will have low affinity and high Vmax. Okay. You know what is the high Km and the high Vmax is? Did you take any lecture before about enzymes? The relationship between the concentration of the enzyme and the concentration of the substrate.
I believe
Yes,
we did.
last year.
Okay, you need to review that. Okay, we don't have time, otherwise I would a little bit explain. Glucose 6-phosphate is allosteric inhibitor for the hexokinase. However, it doesn't have any effect on the glucokinase. However, the glucokinase indirectly inhibited by fructose. And in here, we will see the effect of insulin. There is no effect of insulin, glucagon, fasting, feed of carbohydrates. There is no effect in the hexokinase. However, it can play a very important role for the glucokinase. Why? Because the glucokinase is the place where the glucose degraded, as well the place for the glucose to be stored, okay, as a glycogen. The other important regulator, or the other important enzyme, which is the phosphofructokinase. So first, we will get rid of the allosteric regulation. What are the allosteric regulation? ATP and citrate inhibit adenosine monophosphate, and the fructose 2,6-bisphosphate will induce the phosphofructokinase. Okay, we will get, now we know in terms of allosterically how it's regulated. Okay, then the more complicated thing about the phosphofructokinase is how it is regulated via the insulin and the glycogen, or the glucagon. Oh, not the glycogen, sorry, the glucagon. So when the cell formed a fructose 6-phosphate, okay, formation and degradation of the fructose 2,6-bisphosphate. So why we look at this fructose 2,6-bisphosphate? Because it's an inducer to the fructose phosphofructokinase 1. So I think if I talked as much I talked, you will not actually understand it unless you see the figure. So do you know what is this? This is the phosphofructokinase 2. Okay, this phosphofructokinase 2 is a bifunctional enzyme. What does it mean if it's a bifunctional enzyme? When it is phosphorylated, when this enzyme is phosphorylated, we cannot form fructose 2,6-bisphosphate. So it will be in the inactive form. So in order to activate it, this phosphate has to be removed, okay, in order to activate this enzyme. And activating this enzyme will allow for fructose to be synthesized, for fructose 2,6-bisphosphate to be synthesized, and then activation, the phosphofructokinase 1, which will lead to a change from fructokinase, from fructose 6-phosphate to fructose 1,6-bisphosphate. نرجع أشرحها لكم بالعربي صراحة، لأن أحس لو شرحتها لكم بالإنجليش ممكن تلخبط كثير منكم. إحنا عندنا الآن الأنسولين، تمام؟ إحنا قلنا إنه الفوسفوفركتوكاينيز 1 يصير له ريجوليشن بالأنسولين والجلوكاجون، تمام؟ الأنسولين عادة يكون هاي في حالة إنه والله في جلوكوز كثير في الإكسترا انفايرمنت ونيد تو بي absorbed and hydrolyzed، والخلية تحتاج إيش؟ طاقة. لذلك الأنسولين يرتبط بالريسبتور، بيسوي اكتيفيشن لبعض الإنزيم. هذه الاكتيفيشن لبعض الإنزيم هذه بيقلل الاكتيفيتي حقة السايكلين AMP، اوكي، والسايكلين A and B طبعا احنا السايكلين A and B هو يعتبر زي الكاسكيد. الكاسكيد هذا موجود على السيرفس حق الخليه. لما يصير له دي اكتيفيشن اوكي معناته البروتين كاينيز حقه، لما نقول بروتين كاينيز معناته قابل العمليه، معناته ايش يسوي؟ انزيم يسوي فوسفوريليشن. البروتين كاينيز حقه ما راح يسوي فوسفوريليشن للفوسفوفركتوز 2. اوكي so this will form a this will lead this protein هذا الفوسفوفركتوكاينيز 2 will be dephosphorylated، يعني الفوسفات جروب حقه بتطلع، وهذا راح يخليه اكتف. لما يصير اكتف راح يتكون عندنا الفركتوز 2,6 بسفوسفات، والفركتوز 2,6 بسفوسفات عرفنا انه يحتاجه الفركتوز الفوسفوفركتوكاينيز 1 عشان يسوي اكتيفيشن وننتج وتتم عمليه او تكمل عمليه الجلايكوليسيس. واضحه يا شباب؟ في حاله ان الانسولين هاي ممكن يجي سؤال زي كذا في الاختبار، يقول in a condition where the insulin is high in the cell. اوكي fructose 2,6 bisphosphatase is produced, fructose 2,6 phosphatase is inhibited whatever، او مثلا الفركتوز 6 فوسفات، يعني بجيب خيارات مختلفه، لكن هذا ممكن يكون فيه ايش؟ فيه سؤال في الاختبار. لازم تعرفون ان في تو ستيت. الستيت الاولى اللي احنا تكلمنا عنها اللي هي الستيت حقت ايش؟ حقت الانسولين. هذا طبعا زي ما نقول ايش؟ فيلم كرتوني لها. لو تشوفون انه في حاله فيه الانسولين مرتبط، الفوسفات اللي موجود على الفوسفوفركتوكاينيز 2 will be removed، وبكذا بيكون الفوسفوفركتوكاينيز 2 اكتف، وراح يحول الفركتوز 6 فوسفات لفركتوز 2,6 بيسفوسفات، والفركتوز 2,6 بيسفوسفات راح يسوي اكتيفيشن للفوسفوفركتوكاينيز، وهذا راح ينتج لنا، راح يكمل عمليه الجلايكوليسيس ويسوي لنا ايش؟ فركتوز 1,6 بيسفوسفات. اوكي هذا في حاله الفيدنج والاكتيفيشن للجلايكوليسيس في الليبر. طيب الحاله الاخرى اللي هي لما يكون فيه الجلوكاجون. الجلوكاجون وش راح يسوي؟ الجلوكاجون الاي تي بي راح يصير لها هيدروليسيس عشان تسوي اكتيفيشن للادينايل سايكليز. الادينايل سايكليز او السايكلين اي ام بي راح يسوي اكتيفيشن للاكتيف بروتين كاينيز اي. تمام؟ الاكتيفيشن للاكتيف بروتين كاينيز اي راح يسوي فوسفوريليشن للبارت الثاني. احنا قلنا ان الفوسفوفركتوكاينيز 2 بارت، تتكون من فوسفوفركتوز كاينيز 2 وفركتوز بيسفوسفاتيز 2 بيسفوسفاتيز. تمام؟ فالبيسفوسفاتيز هذا راح يسوي ريموفينج للفوسفات اللي موجود في للفوسفات اللي موجود في الفركتوز 2,6 بيسفوسفاتيت، راح يسوي ريموفينج للفوسفات هذا اللي موجود على كربون 2، وراح يؤدي هذا لوجود فركتوز 6 فوسفات. احنا قلنا قبل شوي في الالوستيريك ريجوليشن لما يصير فيه فركتوز 6 فوسفات وش راح يصير للجلوكوكاينيز؟
راح يقلل من
راح يسوي له انهبيشن. تمام؟ وفي نفس الوقت هذه العمليه معناته ان الفوسفوفركتوكاينيز 1 ما راح يصير له اكتيفيشن وتتوقف عمليه ايش؟ عمليه الجلايكوليسيس. تمام؟ واضحه يا شباب؟
اي نعم واضح.
واضحة، بس دكتور معناته انه الجلوكاجون يعني عشان يسوي انهبيشن
للجلايكوليسيس
ايه، والانسولين يصير له اكتف.
يسوي اكتيفيشن للجلايكوليسيس. ليش الجلوكاجون يسوي انهبيشن للجلايكوليسيس؟ لانه يطلب من الخليه انها تبدا تصنع الليفر يطلب منها يلا صنعوا لنا جلوكوز، لا تسوون له ديجريديشن، صنعوا لنا ايش؟ جلوكوز. عرفت؟
ايه.
الريجوليشن اوف ذا بايروفيت كاينيز الفيد فورورد ريجوليشن باي فركتوز 1,6 بيسفوسفاتيز. لما يصير الفركتوز 1,6 بيسفوسفاتيز عالي، تمام، معناته راح يصير فيه اكتيفيشن للبايروفيت كاينيز، وهذا يعتبر من ايش؟ من الالوستريك ايش اكتيفيشن. الكوفيلنت موديفيكيشن للبايروفيت كاينيز، طبعا هنا احنا بنشوف برضه حالتين، لما يكون فيه الجلوكاجون ولما يكون فيه ايش؟ الانسولين. لما يكون فيه الجلوكاجون بايند للريسبتور، الاي تي بي راح يصير له هيدروليسيس عشان يسوي اكتيفيشن للكومبلكس اللي هو سايكلين اي ام بي. السايكلين اي ام بي راح يسوي اكتيفيشن للبروتين كاينيز. الاكتيفيشن البروتين كاينيز تمام راح يسوي فوسفوريليشن للبايروفيت. تمام، راح يسوي فوسفوريليشن للبايروفيت كاينيز. لما يصير فيه فوسفوريليشن للبايروفيت كاينيز وش راح يصير؟ راح يكون في الان اكتيف فورم. تمام، يعني الان اكتيف فورم حق البايروفيت كاينيز اللي هو الفوسفوريليتد فورم، واللي يتم خلال عمليه الجلايكوجين. وش الانزيم اللي مسؤول عنه؟ الاكتيف بروتين كاينيز A. اوكي، وبكذا راح تتوقف عمليه الجلايكوليسيس. الفوسفوينول بايروفيت ما راح يتحول لبايروفيت. تمام، العكس يصير لما يكون فيه ايش؟ لما يكون فيه انسولين. تمام، الانسولين راح يحفز بروتين، البروتين هذا اسمه بروتين فوسفاتيز. الفوسفاتيز هذا وش يسوي؟ لما نقول فوسفاتيز معناته ريموفينج انزيم يسوي ريموفينج للفوسفات. لما نقول كاينيز معناته انزيم يسوي فوسفوريليشن. الفوسفاتيز بيسوي ريموفينج للبايروفيت، للفوسفات اللي موجوده على البايروفيت، وبكذا راح يسوي ايش؟ اكتيفيشن للبايروفيت كاينيز، وراح تكمل عمليه ايش؟ الجلايكوليسيس، وراح يصير عندنا ايش؟ بايروفيت. اوكي، لما نيجي نتكلم عن الكومبلكس، البروتين كاينيز A يتكون من ريجولاتوري كومبلكس وكاتاليتيك كومبلكس. تمام، لما يكون الريجولاتوري كومبلكس بايندينج للكاتاليتيك كومبلكس هو الان موجود في الان اكتيف فورم. يعني ما هو اكتف. الريجولاتوري كومبلكس ترتبط في الكاتاليتيك كومبلكس عشان ايش؟ عشان تسوي لها انهبيشن. تمام، لما يصير لما يرتبط الجلوكاجون ويصير فيه فوسفوريليشن، راح الريجولاتوري كومبلكس هذه راح يصير لها ريليز، راح تفك ارتباطها بالكاتاليتيك ايش؟ كومبلكس، وبكذا يكون ايش؟ يكون اكتف. طبعا upon its activation it will phosphorylate phosphorylated protein. طبعا في هذه الحاله بيكون عندنا البايروفيت كاينيز inactive، الفوسفوفركتوكاينيز بيكون inactive، والفركتوز 2,6 بيزفوسفاتيز بيكون ايش؟ بيكون active. تمام، وهذه الباثوي تسمى انهبيتوري للجلايكوليسيس، وهذه تكون في حاله ان الجلوكاجون فيه اكشن، يعني الجلوكاجون موجود. الاذر واي يكون فيه الانسولين. الانسولين راح يحفز لنا انزيم اللي هو البروتين فوسفاتيز. البروتين فوسفاتيز هذا يسوي دي فوسفوريليشن للبروتينز، والدي فوسفوريليشن هذا تمام، خلينا نركز، الدي فوسفوريليشن يسوي اكتيفيشن للبايروفيت كاينيز، ويسوي اكتيفيشن للبايروفيت كاينيز 2، ويسوي دي اكتيفيشن للفركتوز 2,6 بيزفوسفاتيز، يسوي لها ايش؟ ان اكتيفيشن. وهذه الاستيت ايش؟ هي الحاله اللي يكون فيها الجلايكوليسيس اكتف. ايه يا سلمان؟
دكتور الحين هذا كله يكون بالليفر ولا بكل الخلايا؟
هذا يكون بالليفر.
وباقي الخلايا كيف يعني؟ ما
باقي الخلايا،
تتأثر؟
هل باقي الخلايا تصنع جلوكوز؟
يعني جلايكوليسيس بس يكون
لا، الجلايكوليسيس يتم في باقي الخلايا كلها، لكن
أي.
البروسيس اللي هي الكومبيتيشن هذه اللي تصير بين الجلايكوجين والانسولين تتم في الليفر. ليش؟ لان الليفر يتم فيه الجلايكوليسيس وفي نفس الوقت يتم فيه صناعه الجلوكوز، فيحتاج لهاي ليفل اوف ريجوليشن. واضح؟
اوكي، اي واضح الكلام.
اوكي، بكذا نكون خلصنا محاضرتنا الثانيه. تبون ندخل في المحاضره الثالثه ولا نوقف؟ اللي يريحكم. يعني انتم الان الحق لكم، تبون نوقف تحسون انه تو ماتش؟ تبون تهضمونه الين بكره وبعدين نكمل؟ اوكي تمام. بكذا احنا يكون خلصنا اول محاضرة